In this video we'll look at how nucleotides are formed and how we make polynucleotides. All nucleotides contain a nitrogen containing base. They have a pentose sugar and a phosphate group. So what we'll do in this first part of the video is we'll look at how they're put together. In DNA, for example, the pentose sugar is always deoxyribose. In RNA, which we'll look at later and you'll look at in this topic, it's ribose. Now this is just an example of one of the four bases in DNA. We'll look at base pairing rules. You might remember them from GCSE, but adenine is one of the four bases in DNA. To make the nucleotide, the sugar and the base join together by a condensation reaction. That creates a nucleoside and one molecule of water as part of the condensation reaction. Then phosphoric acid will join the nucleoside, forming a phosphoester bond between the OH group and an OH group on the carbon 5 of the sugar. That creates another water molecule which forms the whole nucleotide. So now we know the structure and the formation of nucleotides, we need to see how polynucleotides are formed. So through the polymerization we make nucleotides, we make polynucleotides. They are the basis, they are the monomers of DNA and RNA. Nucleotides join together in a condensation reaction, which creates a phosphodiester bond between the OH group on the phosphate group of one nucleotide and on the OH group of the carbon 3 of the other nucleotide. So the phosphodiester bond will form, and remember when we counted the other carbons, 1, 2, 3, so this will form here between carbon 3 of the other nucleotide, and this will probably make it a little bit more clear. We've also got the carbon 5 end, so referred to as 5' and a 3'. So the 3' end of these, and this is sometimes hard to follow, but the 3' end of the nucleotide refers to the end with the carbon 3 at the bottom of it if we're orienting it, and the 5' end refers to the end with carbon 5. So 1, 2, 3, 4, 5. More nucleotides join by further phosphodiester bonds, and that creates the polynucleotide chain. And when you're looking at these in textbooks or in exam questions or wherever you see it, you'll see the 5' end and the 3' end, and that will tell you which way round the molecule is. It's important because you've got an anti-parallel chain that runs in the opposite direction, so that will run from the 3' to the 5' end. It is the same, except it's flipped upside down. So if you were to rotate this the other way round, or just to flip it over from end to end, it would resemble the same structure. Obviously it'd be different bases because it's pairing with the anti-parallel chain, but it just tells you which way round it is essentially. The bases that form are complementary to each other, so you may remember the base pairing rules, but A is complementary to T, G is complementary to C, and the bases get linked by hydrogen bonds, so that's important to remember. It's not so important to go into the detail with it, it depends on your specification, but you will find that A and T are linked by 2 hydrogen bonds and G and C are linked by 3 hydrogen bonds. That is to do with the structure of the bases and the ability for them to form the hydrogen bonds. The next thing to look at with this is to have a look at DNA replication and try and put all of this together.